Okay, everyone, welcome to yet another episode of our Western Evolution Biology series. I'm your host, Matheus, and today I have Joe with me from Florida State University. This is our second episode of season two, and I'll let Joe take it away and teach us a little bit about density-dependent selection. Joe, the floor is yours.
Thank you, Matthias, and thank you all for tuning in and listening and watching this presentation. My name is Joe Travis, as Matthias has said, from Florida State University. I consider myself a population biologist. I try to work at the interface of ecology and evolutionary biology, and my career has uh, taken a variety of turns. I have worked on birds, on amphibians, on a little bit on plants, and the last big part of my career has been on piece-ciliate fishes. So it's for me very rewarding and a lot of fun. So with that introduction, I will begin. But first, actually, I won't begin. But first, let me say that if at any point any of you who are watching this would like to follow up with questions for me individually, please feel free to email me or contact me in any particular way that works. Email is the best, but I would be happy to answer your questions and even chat with you if you would desire. Okay, that said, let's talk about density dependent selection. And let me begin with a slide from a paper published in 1962 by the late Robert MacArthur. This was a monumental paper in which MacArthur demonstrated that given certain assumptions, when a population is regulated, that is, regulated in its density, the measure of fitness that is appropriate is not the rate of increase, which is the traditional population genetic measure, but the equilibrium population size or the carrying capacity, if you will. This was MacArthur's formal demonstration that that is the appropriate measure of fitness and that evolution might proceed differently under such a situation. Well, this was a monumental achievement, and in my mind, is one of the greatest papers in evolutionary biology ever written. It caused an enormous amount of excitement at the time, the excitement and promise of density dependent selection, because it was something that evolutionary biologists and ecologists could both grab onto and hold. On the evolutionary side, papers like Wyatt Anderson's paper showing how density dependent selection could link numerical dynamics of populations and evolution. Charles Worth and Giesel, Demography and Fitness. Jonathan Ruff, now Joan Roughgarden, on the maintenance of polymorphisms. Uh, Brian Clark on, let's see if I can, whoops, on the alleviation of genetic load. Dennis Chitty on the ecological side, regular population oscillations. Michael Bulmer, character displacement. And of course, Eric Pianka and J.P. Grime, life history diversity. Pianka's R and K selection, Grimes' three peers of selection. A lot of excitement and promise. You would think, given the excitement, given the importance of this, that we would be awash in studies of density dependent selection. That's not quite true. This is thank you, Matthias. Uh, this is a graph from Web of Science of citations you can get inserting the term sexual selection or density dependent selection. And this is not quite it's roughly chronological order, but not exactly. It arranges them in order of citations. But the temporal trend is this, of course. More and more citations occur over time. The graphs look very similar, except that if you look at the y-axis, the vertical axis, the sexual selection, at its height has been citations run 1600 to 1800 per year. Density dependent selections running 80 or so per year, an order of magnitude more. So density dependent selection has not overwhelmed the literature like one might have thought it would have back in MacArthur's day. Now, to be sure, there are plenty of examples of density dependent selection. And these are photos of some of the organisms involved in some of what I think are the paradigmatic studies, the great tits, uh, lizards, impatiens, the St. Kilda sheep, etc. And you can find pretty good evidence from natural populations that density dependent selection acts, that fitness depends on population, or selection depends on population density, whether it's long term observations of populations, density manipulations or population comparisons and experiments, there is a good bit of evidence. However, and this is all from the field, however, if you think about the fact that MacArthur's paper was published in 1962, and here is my roster of compelling examples, over 60 years, that's not a lot of papers. It's not a lot of papers. Why not? Well, there have been a lot of papers on theory, starting back in Brian Charlesworth's work in 1971 through Marjorie Asmussen, Charlesworth again around 2013, Why We Are Not Dead 100 Times Over, and a series of great papers by Steiner Engen and Bernd Erik Sather and Russ Landy. A lot of the work has been based in theory. It has proven to be a rich vein of theory. However, empirically, it's not there. And if you look at these big summations and reviews and meta analyses of phenotypic selection, for example, by Adam Sapilsky on temporally varying selection, Joel Kingsolver and Sarah Diamond on another one of their papers on synthetic analyses of phenotypic selection, a meta analysis that looks at something like 2,000 studies, almost none of them really worry about whether selection is density dependent. Well, that's an interesting paradox, isn't it? Here's the thesis for this talk. The density dependent selection is widespread and deserves much more attention, and not just via intra or interspecific competition, because that's the context in which people usually think about it. So the roadmap in which I'm going to argue this thesis has three parts. That's the roadmap for this presentation. First, I'll give you a synopsis of theory with as little mathematics as I possibly can, mostly words. And then second, I'll talk about testing that theory with Trinidadian guppies. That's a major empirical part. And third, I'll offer you two suggestions for horizons for new efforts, where I think new work on density-dependent selection would really change our view of things quite a bit. So let's begin with a synopsis of theory. So fitness is an easy concept to think about, but very difficult to define operationally. But let's take this definition. In an unlimited environment, the fittest strategy is the one that increases the fastest. So the innate rate of increase of a population is a measure of the mean fitness, and each phenotype or strategy's individual rate of increase is its individual fitness. So the fittest strategy is one that this is the classic definition in population genetics. In a limiting environment where the population is regulated, that is to say, increases tend to follow decreases, decreases tend to follow increases, and there is a steady state distribution of population sizes, the fittest strategy is the one that persists at the highest density of the limiting factor. Now, that doesn't sound quite like what you may have expected me to say. The fittest strategy is the one with the highest carrying capacity. And the reason is that this is a very general principle. And we argue this principle in our review paper last year in the annual review of ecology and systematics, evolution systematics. This is the mathematical version of that theory. In a stochastic environment, and this is work based on Shripad Tuljapurkar, Russ Landy, Steiner Engen, Bernd Erik Sather. When density independent fitness, what selection does is maximizes this quantity S, which is the rate of increase discounted by a measure of the environmental variation. And if you want to think in terms of deterministic environments, then environmental variation will be zero. And we're right back to the traditional population genetics measure of the rate of increase. For density dependent fitness, selection maximizes this quantity Q, which is 1 minus a discount based on environmental variance times the rate of increase divided by the sensitivity to the depressant effects of density. And I'm going to come back to that gamma in just a moment. Now, again, if you want to think deterministically, let this be 0. This becomes 1, and we're back to the carrying capacity K in a classic logistic model, which is what MacArthur actually showed. So we can recapture MacArthur's derivation, where fitness is the carrying capacity in a logistic model, the rate of increase divided by the sensitivity to density. So that's the mathematical version of that argument. Now, this theory is agnostic to the ecological source of selection. So, for example, we say the fittest strategy is the one that persists at the highest density of the limiting factor. When resources are limiting, such that you have intra-specific competition driving natural selection and the limiting factor for the population, the fittest strategy drives resources to the lowest level, thereby having the highest population density at that level. That's very similar to Dave Tillman's version of R star in interspecific competition. So the fittest strategy drives resources to the lowest level, a level that it can persist on, but the other strategies, be they phenotypes or genotypes, however you're looking at it, the other strategies can't persist at that level, and thereby our fittest strategy has the highest population density. But it's also true if predators are regulating the population. And I use the term regulating because we're talking about a limiting environment. Regulation means increases tend to follow decreases, decreases tend to follow increases in density, and there is a steady state distribution of density that holds. Here, the fittest strategy is one that persists at the highest predator density, because that's what's limiting. And again, the fittest strategy will be one that will have the highest equilibrium density of its own at the highest predator density. So that's how the theory is agnostic to the source of selection. Now, the theory is also agnostic as to the traits under selection. Notice that this is really theory for what selection maximizes, not how individual traits might be favored at different densities. So, for example, um, when we have situations like this one, and this is a graph of some data from the least killifish, Heterandria formosa, which I've studied extensively in North Florida. Below it is a map of all the places where the data are from which the data are drawn throughout North Florida. And this is based on 12 years of data, the long-term uh, log of density, the characteristic density in different populations. And what this graph shows you, if you look at the top, is that populations at a higher density, females produce bigger offspring. The average offspring size increases. So offspring size is greatest in populations where the density is highest. Offspring size is lowest in the populations where the density is lowest. And because there's a trade-off between offspring size and number, the average fecundity, the amount of offspring that a female will produce in her lifetime, varies inversely with density. The open and closed symbols denote different types of habitats, springs versus ponds, but you can see these all follow the same pattern. This is the kind of observation that got ecologists excited about density dependent selection, because ecologists thought hard in the 60s and 1970s, and some of us still do today, that the traits that contribute to fitness when a population is at low density could be different than the trait values that contribute to fitness at high density. This became the impetus for a lot of work on density dependent selection. However, the theory doesn't really draw the contrast between low density and high density. It draws a contrast between unlimited and limiting environments. And so to apply the theory in this case, you'd have to say, well, at very low density, perhaps these are not a limited, perhaps this is an unlimited environment, and these are a limiting environment. But the theory is agnostic to the traits. Predicting the evolution of particular trait values under density dependent selection requires detailed demographic models. And the top two panels, which I'm not going to walk you through, I just want to show you what it looks like, are from a paper by Tim Coulson et al. In 2022 in the peer community of evolutionary biology. I am a co-author on this paper, and it is a uh, examination of how density dependent selection could select for smaller or larger body sizes. The same basic principle that the evolution of particular trait values requires detailed demographic models was articulated beautifully way back in 1988 by Larry Mueller in this paper in the American Naturalist. And what Larry showed there, and what we tried to show many years later, is that in density dependent selection, you can get a variety of outcomes that depend upon for traits depend on the particulars of the model. So That means that our simple definition of density dependent selection, drawing a simple contrast between unlimited and limited environments, hides a rich diversity of theoretical possibilities. Well, that's, and again, I emphasize to you that the important distinction is between populations in an unlimited environment and populations in a limiting environment. Well, that was my synopsis of theory. I want to talk about testing this theory with Trinidadian guppies, uh, which is work that I've done in conjunction with David Resnick, Ron Basser, Tim Coulson, Andres Lopez Sepulcre, and others over the years. So let's test the theory with Trinidadian guppies. This is a Trinidadian guppy. These are apicellate or live-bearing fish. They occur throughout the island of Trinidad, but I'm going to talk about studying them in the northern range mountains. If you're thinking in how big these fish are, the females are probably about an inch. At most, the males are probably 3/4 of an inch, perhaps an inch for a really big male, an inch and a quarter to an inch and a half for a really big female. But that's a really big fish. In the mountains, northern range mountains of Trinidad, the rivers look very different at the higher elevations at the lower elevations. Now, I should point out at the higher elevations, we're only talking about uh, 500, 600 meters at most. Well, I guess the highest point is at 900 meters, but we're really looking at streams that are about 500 meters or so. At the top, they exist as a series of pools and riffles that look like this on your left. At the bottom, a more conventional river, a broad, flat, and continuous. The bottoms, the lower elevations, are filled with a diversity of piscine predators. This Cranicycla on your upper left, a wolffish, Hoplius here on the upper right, this flock, as if they were birds, school of Astyanax here, uh, very, very high predation. As you walk up the river, beginning at the bottom and moving up the steep mountainside, you encounter a series of what we call barrier waterfalls that form when calcium compounds precipitate out of the water with an abrupt elevational change, and these form a barrier to dispersal. The larger fish cannot get over the barriers. The smaller fish go around the barriers during flooding. And in this slide from a marvelous paper by Jim Gillum, you can see what happens at different sampling stations as you walk up the Guanapo River. And here on the bottom, on the axis is the distance from the first sampling station, the elevation in meters. And you can see they're only about 300, uh, between about 300 and 800 meters here. And you can see how steep the mountain sides become. And these are predators like Cranicycla, Aquidens, Hoplius, the wolffish, Astyanax. Notice how they drop out at different elevations, depending upon the species. And by the time you get to the highest elevations, guppies are found throughout. You really have only guppies and Hart's killifish. Ramdia um, occasionally gets up that high. So you have a situation in which you have a continuous variation in the intensity of predation going from low elevation to high elevation. This pattern is repeated throughout the Northern Range Mountains. These are the two drainages of the Northern Range Mountains, the Caroni on your left here, the Oropouche on your right. These are sampling stations that we looked at along the different streams feeding into each drainage. And what I want you to see, we characterize them as high and low, but here this in the Aripo River at a more thorough sampling, we characterize them as different intensities of predation. But you can see by the coding of the colors, there's continuous variation in fecundity and offspring size. From a situation at the bottom where females are very fecund, on average about 7 offspring, each one weighing 0.8 milligrams, to the top where they produce very few offspring at a time, 2.3, but each one is really big, twice the size of the ones at the bottom. This continuous variation is characteristic of every stream. At the extremes, the very bottom and the very top, where they occur only with killifish, the males look like this. And we call these high predation and low predation. We know from population genetic data that fish at the low predation, higher elevations are descended from colonists that came from the high predation lower elevation. So colonization moves downstream to upstream. But I want you to notice well, there are three things to notice here. One is that you don't need any fancy statistics to tell you that the males on the right are much bigger than the males on the left. The second thing you'll notice is that they're much more colorful on the right than they are on the left. And that's gotten a lot of attention from a lot of people over the years. And I'm not going to talk about the coloration today. That size is interesting because male guppies do not grow after they mature. And that means that these guppies matured on the right at a much larger size than the ones on the left. And in all piece-ciliate fish, the size at maturity is directly proportional to the time to maturity. So that also means that because they are so much bigger, they matured much later. They delayed maturation in order to achieve that size. That's the issue I want to address, this delay in maturation, which you would think makes, well, it would make very little sense down at the bottom. Why would you delay maturation? If there's high predation, your life is going to be nasty, brutish, and short. You should mature as early as possible. Theory suggests, however, when that is not the case, when you can expect to live quite a while as an adult, and there is a fitness benefit in getting large, you should delay maturation till you reach a certain size. So this looks, at least at first glance, I'm sorry to flip this pictures for you. Uh, this could be high predation, low predation. Do predators select for this? And this, by the way, is what's actually in a lot of textbooks on evolution, that the alleviation of predation pressure has selected for later age and larger size and maturity. However, as any good ecologist knows, if we, and as I we make a graph of predation pressure on the horizontal axis and average population density on the vertical axis, and you look at a bunch of different populations, you're going to see a very nice inverse relationship. Places where predators are more abundant and predator pressure is highest, that prey population will be at much lower density than when predation pressure is low. So the low predation, high predation is actually a contrast between two very important ecological forces, predation pressure and population density. Because up at the top, where there is almost no predation pressure, population densities are much higher, food is much more limited, the diets have changed to reflect that. And so we can ask the question, which is it? And is this an example of density dependent selection theory, where at the bottom, they are not in a limiting situation, because, by the way, these predators eat all kinds of things, not just guppies. And at the top, they're in a limiting environment. Well, what would it take to falsify the predation hypothesis and support the density dependent selection hypothesis for delayed maturity? Theory tells us we need to do three things. First, demonstrate that there's density limitation in our low predation populations. Second, demonstrate that the density limitation occurs through recruitment, some combination of adult female fecundity and juvenile survival. If density limitation was occurring through adult survival, that would not select for delayed maturity. I don't want to go into the theory, but I will just tell you that's the result. So we have to show that it's recruitment, not survival. And finally, we have to demonstrate that the trait change does not begin to occur until the population density approaches a steady state situation. And here I have to go back to what I told you earlier, that the high elevation, low predation populations form after colonization by a high predation, low elevation phenotype. So they colonize bottom to top, and the trait change should not begin to occur until population densities have gotten high enough to approach that steady state distribution. Because at very low densities, we shouldn't see the delayed maturity begin to evolve. If it were predation, it should start to evolve immediately. Well, let's take these one at a time. Density limitation in low predation populations. The pool and riffle structure offers a great opportunity to study this because the guppies really don't exist in the riffles. They don't like being in the riffles. It's very shallow, water moves fast. They aggregate in the pools. That means we could actually manipulate the composition of pools by putting little barriers upstream and downstream of the pool and using these as experimental arenas. We can do this and increase the density of guppies in some pools, decrease the density in other pools, leave some pools unchanged, and see how that affects population growth rate over a 28-day period. And here's the result. Experimental increases or decreases in density in our pools either reduce or elevate population growth rate. These four panels illustrate four different times we did the same experiment, manipulating density. And I'll just look at the upper left for the moment. We start with 1.0x, meaning the actual density of the pool. In pools where we halved the density, population growth rate went up. In pools where we increased the density by 50%, it went down. Where we doubled the density, it went down even more. Same was true in a different drainage here. Where we leave the density the same, population growth rate is the same. Lambda is 1. Population holds steady. Decrease the density by half, it increases above 1. Increase the density, it decreases below lambda equals 1. Same in this different set of experiments with a different design. So, experimental increases or decreases in density reduce or elevate population growth rate. There is density limitation in these populations. Second, can we demonstrate that it occurs through recruitment? Now, a 28-day period is not a very long period to really estimate recruitment, though we do have evidence that his recruitment was affected by those manipulations. We really would like to see this in a much longer time series. Our solution to this problem was to recreate the colonization of an upstream location. We transplanted high predation guppy stock upstream into reaches without guppies, but with the killifish that are normally there in a controlled experimental design. In other words, we captured juveniles, raised them to adulthood, took them up across the series of barrier waterfalls, and released them in experimental streams upstream. So this recreates the natural process of colonization, albeit, they colonize much faster than they would have made their way up otherwise. Here's where we did it in a stretch of the 4 different reaches in the Guanapo River drainage. Here's the island of Trinidad, the enlarged area here in the Northern Range Mountains. Here's the Guanapo River. And here are our 4 experimental populations, the Taylor and the Kaiwa, what are called upper and lower Lalaha. These populations were introduced in 2008. These were introduced in 2009. Here we are dropping the fish into one of the pools. Every month we survey these fish, so we do mark recapture. Our recapture rates are something like 90% to 95%. Guppies, while the males move a good bit, females tend to be have a lot of site fidelity. So we get high recapture rates and excellent estimates of the demography of these populations, meaning not just population densities, but the distribution of individuals among the different size and age classes. Here's a graph of the biomass density, grams per square meter of guppy in our 4 experimental populations through the month 103 back from when we started. And this is the month. The solid lines are, the blue lines are the upper and lower Lalaha. The red lines are the Kaiwa-Taylor pair. The solid lines are reaches in which we did not thin the canopy. The dashed lines are reaches where we thinned the canopy. That's an ecological manipulation I'm not going to talk about right now, but in each pair, we thinned the canopy in one and kept the canopy intact in the other. The thinned canopy allows a lot more light, higher productivity, and you see these oscillations in density that correspond to big increases in the dry season in each of those streams, much more exaggerated compared to the changes in the intact canopy streams. But we have these population densities. From these data, we can look at the steady state distribution of size and use what are called state space models of the monthly change in the log of density to show evidence of regulation. These data were published last year in a paper in the American Naturalist. And what these graphs show you is that decreases tend to follow increases more often than you'd expect by chance alone. Decreases tend to follow increases more often than you'd expect by chance alone in all four populations. So these state space models take the actual observed data, correct for the experimental or sampling error, and give you confidence limits and estimated values. So they show evidence of regulation. This is a graph of the monthly recruitment, the number of new juveniles captured, graphed against the population density in the preceding month. And you can see that in each of our four streams, this tends to be an inverse relationship. Now, these are the raw data. If you adjust these data for the seasonal oscillation in recruitment, you get a much cleaner relationship, and that's described in the paper. But I like showing the raw data so you can see that even in the raw data, without a lot of statistical manipulation, there's an effective recruitment. There's no effective adult density on survival. So we've now fulfilled number two, demonstrate density limitation occurring through recruitment. What about number three? How are we going to do that? Well, uh, let's take a look. Here's the expectation. This shows you the mean phenotype, that is the age of maturity that we should see. If predation is actually the agent of selection, as soon as we alleviate predation, the mean phenotype should begin to change. In this case, maturity should be beginning to be delayed. The age of maturity should get larger immediately because we've alleviated predation, putting them at low density, and that's what we expect to see. The dashed lines are the expectations that there's density limitation. There should be a lag depending on the initial length of the lag, depending on the initial density. These are from models described in the Resnick et al. 2019 paper in American Naturalist. There should be a lag, and we shouldn't see the age of maturity begin to increase until sometime afterward. This bottom graph shows you the selection gradient. If it's predation, selection should be most intense at the very beginning, favoring a delayed maturity. Under density limitation, it should not be very intense because they'll still be at the very low densities characteristic of the source population at high predation. So the selection gradient should steadily decrease under predation. Under the predation hypothesis, it should steadily increase under the density hypothesis. But the real key is the mean phenotype. So this makes sense. All we got to do is go out there and measure male size. Well, that's not quite right because size and age and maturity are phenotypically plastic. And to factor out the effect of plasticity, we have to do common garden experiments. So that means that for every year, we collected juveniles from all of our four populations, plus the high predation location that we used as our initial stock, brought them back to the laboratory in the United States, raised them for 2 generations, and assayed the age and size and maturity of males in a common environment so we could eliminate the effect of phenotypic plasticity. And here are the results. The annual common garden experiments on F2 fish, that is, we brought the juveniles back, raised them to adulthood, raised their offspring, and then measured the next generation, so the grand offspring, the F2 fish. They show the predicted delay in evolutionary change. So I want you to look on the left. This is the difference in the age at maturity from the control, from the high predation stock, in the upper and lower Lalaha. The top panels are the age of maturity, the bottom panels are the size of maturity. I just want you to look at the top panels for now. Notice for the upper and lower Lalaha, there was no difference from the controls in 2009. And these populations were started in 2008. There's no difference in 2010. 2011, they're starting to get delay maturity, males by an average of 5 days. By 2012, they're delaying maturity by an average of about 12 days. In the Taylor-Kywall pair, which were started a year later in 2009, 2010, they are no different from the controls. 2011 and 12, they're already quite different. They achieved the delay began, the lag here was 2 years, the lag here was 3 years. These fish, these populations were initiated at a lower initial density than these fish. They reached the threshold density in Taylor and Kywall sooner, a year sooner, than they did in the upper and lower Lalaha. And this is described in detail in this paper, Resnick et al. 2019 in the American Naturalist. Further, Here's the difference in age at maturity in all the populations graphed against the biomass density of their grandparents. So the change in mean phenotype was associated with a change in with the density in the grandparents' generation, strongly consistent with exactly what we would expect for the theory for density dependent selection. Well, I've given you a synopsis of theory. I've showed you how we I've shown you how we test that theory with Trinidadian guppies, and I want to offer you two suggestions for new efforts. Now, the first looks back at some of these great demonstrations of density dependent selection in the wild. Great tits, impatiens capensis, etc. They took advantage of this theory. Now, I mentioned at the very beginning of this talk that when MacArthur did his work, he made a few assumptions and made this elegant demonstration. The same assumptions are made in these papers by Tulchbekar, Russ Landy, Steiner, Engen, and Sather. That important assumption is that competition under density limitation is symmetric. Now, this is for a situation of resources being the limiting factor. Intraspecific competition is symmetric. And under symmetric competition, you can derive this theory. What does that mean? Symmetric competition means that each genotype competes equally against every other genotype. This is a simple model drawn from Marjorie Asmussen's paper. I mean, it looks like a lot of symbols, but it's very simple. It's a linear model for her paper in 1983, where the fitness of each genotype is a function of its innate rate of increase, the W with the W1, W2, W3 for these three genotypes, and then these competition coefficients against other genotypes. So alpha 1, 1 here is the effect of the A1A1 genotype on itself. The alpha 2, 2 is the effect of the A1A2 genotype on itself. The alpha 3, 3 is the effect of the A2A2 genotype on itself. And this is based, her model, well, in this particular model, she let these things vary. So in symmetric competition, all of the off-diagonal alphas would be equal to each other. They would all be equal to each other. So that the only thing that really differs between the genotypes is their own sensitivity to the depressant effect of density. That recaptures the standard theoretical result, as I showed you. The fitness maximizes or selection maximizes this quantity Q, the rate of increase divided by the sensitivity of the depressant effects of density. And you can get a comparable expression for different types of density relationships, not just logistic. You can get one for a Beveridge-Holt relationship, for example. So that means that when there's symmetric competition, we recapture the classic result. Under asymmetric competition, when each of these off-diagonal alphas, when they are not equal, when they are all different, no single quantity is maximized. So the outcome of density-dependent selection, sorry about that, that's a typo, can be predicted only via invasion analyses. In other words, you cannot simply look at the fitness of an individual genotype under a range of densities and simply predict the outcome of density-dependent selection under asymmetric competition. Well, let me show you what that means. We did this experiment with guppies. We compared the competitive impact of high predation and low predation guppies on each other in a laboratory experiment. And this is from Thomas Potter et al. 2019. This graph shows you competitive equivalence on the vertical axis as a function of the body size of a low predation guppy and a high predation guppy on these axes on the bottom. And this equivalence shows you how many high predation guppies it takes to have the same competitive impact as a single low predation guppy. Now, it's based on body size because competition is size dependent. So this is how many high predation guppies of a particular size does it take to equal the competitive effect of a low predation guppy of a particular size. So just for fun, let's look at the far back corner up here, where we have a 10 millimeter long high predation guppy and a 22 millimeter long low predation guppy. And it takes about 3¼ high predation guppies to equal the effect of one low predation guppy. Now, you're not surprised. This is a little fish. This is a fish that's twice as big. It ought to take a lot more of these than these. But notice that when we start moving away from the ridiculous extremes, notice that the plane that describes equivalence is almost always above one. And that the only time it's really below one, that is to say, the high predation guppy is the better competitor, is really for very large high predation guppies and small to moderate low predation guppies. In other words, most of the time, the low predation over most of the range of body sizes, the low predation guppies are much stronger competitors than the high predation guppies. So competition is asymmetric. Guppies are not the only example. Asymmetric competition has been demonstrated in lizards, in paramecium, Leo Luckenbill's old experiments, in Drosophila, in pitcher plant mosquitoes, Bill Bradshaw and Christine Holzapfel's experiments. Yet, yet, in studies of density dependent selection, most of them, frankly, do not investigate whether competition is asymmetric. Excuse me. So when we look at density dependent selection based on resources, we still have a great deal to learn about the importance of asymmetric competition. This is a largely uncharted area. There are other studies than these, by the way, that I'm showing you. An elegant study by Janis Antonovics and Norm Elstrand on grasses. But while there are other studies, they are a small roster of studies compared to the possibilities. My second horizon is in sexual selection. And when we think about sexual selection, we almost always think about traits like ornaments in males, body size differences, different male phenotypes, and sometimes characteristics of sperm, as in these red sea urchins, the velocity of sperm, the longevity of sperm. We think only about the effects of males, and that makes sense if you think about it. Um, there is a history of density dependent mating and sexual selection. There's several great studies on density dependent male allocation and tactics showing that uh, how males, what tactics males use to obtain mates can change with population density, and that at high densities, a different phenotype prevails than you see at low densities. And there are very nice studies showing density dependent sexual selection, where the actual prevailing male phenotype changes with density. So while these show phenotypic changes, these actually show you sexual selection. Well, this has been well known for a long time that sexual selection can be density dependent. And this is reflected in the fact that sexual selection is usually considered soft selection, where the fitness of an individual depends upon the identity and numbers of other individuals. So, whatever tactic a male uses, its success could depend on the games that the other males are playing, if you will. So, selection is density and frequency dependent. That's the classic definition of soft selection. In sexual selection, male fitness may depend on a male's competitive ability or his attractiveness, which in turn depend upon other males. And when males are not limiting, then all ova will be fertilized. This kind of density dependent selection does not feed back onto numerical dynamics or population size. And this is the way people have thought about density dependent sexual selection for a long, long time. We think about sexual selection as being involving a male's relative fitness, not his absolute fitness. How many mates does he obtain compared to other males? Without much feedback onto population dynamics, because males are not limiting and all the ova will be fertilized. But there's a context in which this might not be true. Uh, Sexual conflict, hard, soft selection, and sexual conflict. And features that enhance male competitive ability via soft selection can inflict harm on females. This is a graph uh, produced by my student Sarah Kettlecamp, former PhD student, in which there are a number of genetic lines, what are called the DGRP lines of male Drosophila melanogaster. This is the males on the horizontal axis and the mean fecundity of mated females from a standardized stock. And it's well known that Drosophila semen contains a number of compounds that can be harmful to females and reduce their fecundity. The traditional interpretation is that males employ those tactics to reduce a female's fecundity if she mates with another male after she mates with him. But what I want you to see is that some male genotypes induce a lot more harm than others. So look down here, this particular genotype, female fecundity is is a third of what it is when she is mated, when a female is mated to this genotype over here. So sometimes the features that enhance male competitive abilities, and these these are more competitive males, uh, can inflict harm on females. This sexual conflict. This has been studied actually in theory by Hannah Coco and Rob Brooks. And what they show is that when this can happen, when features that enhance male competitive ability inflict harm on females, as the alleles for a more preferred male go up in frequency and male competitive ability increases, so does the harm on females. Now, when female fecundity is high, this kind of effect that I showed you in the last slide, females can withstand the harm from the favored male effects. It's not a big deal. But when female fecundity is low, the increasing frequency of the harmful male can reduce female fecundity and carry the population to extinction, what Coco and Brooks call evolutionary suicide. The general point being that sometimes the very males that are favored by sexual selection could induce, which is density dependent, could induce a change in population dynamics through the pleiotropic effects on the harm they inflict on females. And to show you this is not simply a function of Drosophila, I show you this graph from studies of mosquito fish done by my former graduate student, Lisa Horth. The same principle. This shows you female density in experimental mesocosms, large cattle tank populations, as a function of the frequency of melanic males and typical males, what are called silver males. This is a genetic Y-linked genetic polymorphism. And what I want you to see here is that the average female density is much lower in the solid lines. This is the population with the high frequency of melanic males. Than it is in the two dashed lines, which are two sets of experimental populations with a low frequency of melanic males or no melanic males. And the dotted line has no melanic males, the dash some melanic males, the solid line's high frequency of melanic males. Notice that the females were decreasing here, even when the melanic males were not initially high frequency, and that's because they were increasing in frequency. This appears to be kind of a stable polymorphism, but when you have melanic males, there is a lower density of females and a lower population density. This is, by the way, largely an unexplored area. Well, I've given you a synopsis of theory. I showed you how we could test it with Trinidadian guppies. I've offered some horizons for new efforts. And I've argued my thesis that density-dependent selection is widespread and deserves a lot more attention, and not just in the context of intra or interspecific competition. It is a very general theory, it's a very profound theory, and I think it deserves a lot more attention. And there are some areas that are relatively unexplored. Well, I want to finish by acknowledging a lot of people who have been involved in the Guppy project over these last 16 years or so that we've been doing it, since really 2007, 17 years. This shows you a roster of some of the principal actors, color-coded by their roles. So the people in gold, Ron Basser, Tim Coulson, David Resnick, and myself, are the PIs who are still actively working in Trinidad. We have people in blue that were PIs on the original study way back when who have moved on to other things. We have our graduate students and postdocs in the orange color, and we have a lot of our field managers in green, Mark Charon, Camille Chia, and Annie Van Alst in particular. A lot of people contributed to this work. We could not have done this work on guppies without the large team over the 17 years, and they deserve plenty of credit. So that's my story. I thank you for listening, and I know Matthias and I will chat a little bit. But if anyone watching or listening has questions they want to ask individually, please feel free to contact me. My email is Travis, my last name, at bio dot fsu dot edu. Thank you very much. So Matthias, I'm going to turn it over to you and stop sharing my screen.
Wonderful. Thank you so much, Joe. Just a note on, you shared your email, and I'll make sure as well to tag your email onto the YouTube video
Okay, good.
underneath it. Then folk can reach out to you, because I'm sure they will reach out to you. I must say, thank you very much. This has been a wealth of information. During all your talk, I was thinking, wow, this is a master class. So, I
Thank you
really
so much.
appreciate your efforts, and also in the structure. I really liked the way you structured the talk, particularly with the historical context. I think we oftentimes forget to look back what folks have done before us, and I think we should be particularly attentive to what other folks have done before us in evolution, because evolution itself is a historical discipline, and so much has been done, and we should look back in time and see what has been done before us. So thank you for that.
Sure.
Now, I must say, during the talk as well, I was, grabbing my seat here and thinking, when is he going to talk about phenotypic plasticity? So I'm happy you brought that up. And I was also intrigued with the asymmetric competition, because when you touched upon, for instance, in the experiments you guys did with the pools, right, in which you changed the density of the populations, I was thinking, well, I suppose when you increased the population density in those pools, you brought, individuals from different pools, right? So from different populations. And then I thought of, I did not have the name of asymmetric competition, but then I was thinking, well, if these individuals are coming from different populations, what are they bringing to the table that is,
Right.
inherent of their own populations?
Yeah. What we did, by the way, with those experiments, we actually collected all the guppies and mixed them randomly
Okay.
and then put them back into the pools. And we had to make a decision, because one of the things when you do that kind of experiment, you say, well, should we put them in a range of densities like 10 guppies, 20 guppies, 40 guppies? And we decided to go with fractions of their original density, because we assume, by the way, that each pool might have a different equilibrium size.
Mhm.
And so by having and doubling, what we're doing is looking at population density relative to what was there at the start, relative to the control. So
Mhm.
rather than use absolute numbers, we used relative numbers, and we had quite an argument over how over that.
I can
I
imagine.
think the results prove that's the right decision. But when you're staring at the pool, and well, actually, when you're actually contemplating this, when you write your grant proposal,
Mhm.
um, you're arguing over how to do it, and it didn't take long in the field
Mhm.
when you walk from pool to pool and you see this pool has a lot of guppies and that pool doesn't have so many guppies, you think, you know what? Let's not do it that other way. Let's do it relative to the pool. So a lot of I think what's one of the things that's important is there are a lot of decisions that go into these sort of experiments that you could have done them a different way. And
Absolutely. And that
of course,
shows.
I only show the ones that worked out well, not the total absolute dismal failures. We just think, God, we shouldn't
Rightly
have done
so.
this.
Rightly so. No, and I agree. And that shows that details are important. And speaking of the work in the field, I was just curious, because again, I don't do field work. How do you cited a very, to my opinion, a high recapture rate, right? 95%, I think.
Yep, yep.
And how
50
do
months.
you
Yeah.
guys label or mark the guppies?
So we mark the fish by using elastomer, which is a tiny little bit of plastic. It's kind of a gel-like plastic, and we inject the elastomer under the skin.
Okay.
And we have 7 colors and 8 positions on the fish.
Mhm.
And so, with anywhere from 1 to 2 marks, we can identify every fish uniquely between color and position. And so that's how we know who that fish is.
Very nice.
And it takes a lot of bookkeeping. So, for
Mhm.
example, when we don't see a fish for 6 months, we assume that fish is dead and that mark is free to be used. However, We don't reuse that mark in the same stream from which that fish was living. That mark gets assigned to a different stream. And so every month, someone has to process the marks, see who's gone, and give a list of new marks to administer in
Fair
each
enough.
stream. It's a task in and of itself quite demanding.
Yeah, and that's actually a good point, because from remembering the conversations I have with other ecologists here in my department, I think the discussions around, especially thinking of migratory animals, like migratory birds, right? The discussions about when do you consider whether a bird never returned because the bird is dead or not? And so yeah, I wouldn't have thought of what you said. You need to make sure that you don't use that particular label in the stream anymore. Otherwise, you have a confounding effect there.
Right.
Thank you. And speaking of, again, out of curiosity, as we are discussing details, my understanding is that, guppies are a highly tractable model for various reasons, right?
Mhm.
And you showed us during your talk beautifully how that is so. Now, thinking of next steps, what other groups, species, populations you would see as highly tractable models for the study of any aspect of density dependent selection? You listed a few species already that have been used, but what do you think of would be the hotspots in the future, let's say?
Well, I mean, I think any number of herbaceous plants would
Mhm.
be excellent subjects. I mean, part of the issue with all of this, you need to really look at the evolution of particular traits. You need detailed demography. So you really need to build life tables, and that requires you have some certainty in period to period censuses. And the beauty of plants, as John Harper once wrote a long time ago, is they stand still and wait to be counted. And so they are very well suited for this sort of work. There are a number of animals with good site fidelity that would be suited for this. I mean, we're very fortunate in that we know that some of our guppies that we record as dead probably went downstream.
Mm,
We know that because we
mm,
have done work. It's a beautiful paper by Sebastiano De Bono in Ecology Letters in 2019, looking at movement patterns in these streams, and some other work by my postdoc Tom Potter that was published in Science last year, looking at patterns of movement between males and females. So
mm,
we know they move a little bit, um, and we also know they get washed out, but
mm,
probably killed in flash floods in the wet
mm.
season.
Fair enough.
But the numbers are low. We really don't see when we don't see a fish for 6 months, we can pretty much be sure that it's dead. Or if it's not dead, it's gone and isn't ever coming back.
Is not having an effect on the population. Yeah.
So essentially, it's dead from the point of view of its ecological and genetic impact on that population.
And speaking of herbaceous plants, I'm assuming,
So site fidelity is a big important thing. Unless
yes.
you can do it in a meta population context where you can see how far individuals go and track them
Mm.
through their lives, even when they go elsewhere.
Or
That's
perhaps doing
that's
some sort of
difficult.
mesocosmos array, like
Yes.
you keep them uh still, let's say. And speaking of herbaceous plants, their advantages of using them, perhaps they would also be highly tractable in teasing apart which traits are under the influence of density dependent selection,
Yeah.
right? Because you showed that table in which you really said, traits, they actually have different effects.
Yeah,
And I'm
because you can
thinking,
actually you can get a good handle on adult fecundity. How many seeds does a
hmm,
does a plant produce? You can get seedling survival in many cases. You can get adult survival. So obviously you want to look at perennials.
hmm, yeah.
But you can get it. You can get at these. You can get at those factors very easily in plants, and even in annuals. If you have annuals that continually flower and that can be censused week to week to week to week, you can get the data you need to test theory.
Mhm. And then out of curiosity, because you started off saying that, and you showed us, the bibliometric data showing that sexual selection is sexier, and we have more studies about it. And it seems as though density selection, um, it has been, let's say, overshadowed to a certain extent. So And I was wondering why that is the case, because I agree with you. You know, when I think about, for instance, the courses I've taken, both as an undergraduate and a graduate student in evolution, um, you know, I feel like very rarely it's almost like we speak of density dependent selection as an appendix, you know, oh, this also
Right,
happens, it's
right. It's a,
important, but it's very like, you know, mathematically saying it is thick. Now,
yeah.
do you think that might be the case because, and I'm generalizing here, do you think that might be the case because perhaps as a group, biologists are afraid of math? This is myself,
Um,
you know, because I'm
you
thinking
know,
of my own self and my
I think it's
friends.
generally true that people are afraid of math.
True, true.
I mean, unless you're a physicist or computer
Yes.
scientist or mathematician or certain type of, I mean, people are afraid of math. So I mean, I think there's some truth to that. Um, and I think it's there's truth to that, and I think there's also a tendency for ecologists to think about density dependent selection as something like R and K selection.
Mm,
Then you've had papers that say, well, this was not a very good idea. That's right. So people dismiss it as, well, that was a good idea. That seemed like a good idea, but it's wrong. Well, I mean, there's more to it than R and K selection.
mhm,
And once you get into it, you realize why that paradigm was misleading. And I think it misled a whole generation of ecologists, frankly, who really didn't appreciate the theory in detail. I
mhm,
think there's a certain extent people are afraid of math. And perhaps we as faculty members don't do a good enough job of explicating that math and
mhm,
distilling it into concepts that the students who are not heavy duty mathematically inclined can
mhm,
appreciate.
mhm,
Um,
yeah,
perhaps we're not doing a good enough job. So
mhm,
yes, there's a math phobia, but perhaps there's also a um, failure to really explicate it well. I
mhm,
think there's also a divide between theoreticians and empiricists that's unhealthy.
mhm,
I think too many empiricists are what I call atheoretical, like agnostic, or with an A in front, meaning they don't want to hear about it.
mhm,
I think that's a terrible mistake. I think theory is so vitally important, and I'm really not a theoretician at all. I'm not a theoretician.
mhm,
I'm an empirical biologist, but I was trained, and I was very fortunate to get this training when I was very young. I was trained to think of theory as vital and essential,
mhm.
because without theory, we wouldn't know what to measure, we wouldn't know
Assumption,
what to
yeah.
test.
Yeah.
So I think there's been perhaps a too great a divide
Mhm,
and too great a um, willingness to walk past theory and say, well, that's for those guys over there,
mhm.
rather than, oh, well, this is going to tell us what to measure and what not to waste our time on measuring, and how to make the right inference for the right reason. Yeah,
Yeah,
it's
I
my own
agree
thinking.
with you. I think I see that as well, you know, again, from conversations I have around. And to tie things up, to wrap things up, I must say that again, my hope with this series really is to create that conversation, right? To let folks, you know, give them the opportunity to think about these things we've discussed, not only the topic of the talk itself, but these behind the scenes detail, if you will. And I must say that again, your talk has been very informative, and I'm more than
Thank you.
sure that it will, you know, spark conversations um, about density dependent selection and evolution as a whole. So thank you very much. This was very informative. And I will certainly, you know, watch again to let those concepts sink in. And I'm sure there will be lots of viewers and they will reach out to you. So thank you very much, Joe.
Sure.
I
Thank
appreciate
you,
it.
Matthias. My pleasure.